Hey, hello, everyone. Why don't we get started? It's 10 o'clock hour Pacific time. This is the Human Islet Research Network, or HIRN, webinar series. Thank you for joining. And today we have a really interesting, exciting talk by Dr. Yuval from the Hebrew University in Jerusalem. He's going to talk about extensive cell turnover during postnatal pancreas growth. I'm Joyce Nyland. I'm the chair of Discovery Science Department at City of Hope, and also co-PI of the HIRN Coordinating Center, along with John Cadiz. And I'll just give you a brief introduction to his talk today, and then we'll dive into the material. So just those of you who are not familiar, to tell you a little bit about the HIRN configuration and network, which is evolving continually. Currently, there are 5 consortia listed here, looking at cell death, immunity, biomimetics, and we are the HIRN Human Islet Research Enhancement Center, or Coordinating Center. But this configuration is about to morph. In the future, there will be a new initiative, which I'll tell you more about in a moment, called the Consortium on Modeling Autoimmune Disorders. And we also have just had funded the PANC-BASE Consortium, which will look at integrating type 2 diabetes data across the nation and type 1. And there are new consortia RFP that is out for this CMAD. I'll show you that in a moment. But this consortium and set of consortia have been going on for 9 years now with 800 researchers. There's over 200 currently active, involving 109 institutions across 12 different countries. And if you want to know more about NIH funding opportunities like the new CMAD, you can go to our HIRN newsletter as one source if you'd like to subscribe. And you also learn about new HIRN members and manuscripts, webinars such as these. Job opportunities are posted there, both academic and industry, and funding initiatives such as CMAD, which is due March 20th, 2024. If anyone is interested in applying who hasn't heard of this yet, NIDDK intends to commit $5 million to fund 4 to 6 awards and form this new consortium on modeling autoimmune disorders, and diabetes. If you could take a look at that. And another place you can find out about these funding opportunities and other items is on our website, which is listed here. And here's the funding tab, where we tell you about various announcements and resources. Also, the resource browser, I always like to emphasize that, because it really is a great set of resources for you as a diabetes researcher. And you can follow us on various social media and get more information that we put out there routinely. So today's paper is shown here. Dr. Dorr is going to talk about this cell reports that just came out very recently. And he has inside this, he and his co-authors, a really wonderful, rich resource table that lists all of the antibodies and models and cell lines that they utilized in the paper. And so what our staff do in the HIREC is we've gone through and curated and put all of this information into the resource browser, which is shown here. We now have over 2,800 various resources cataloged in this website, from protocols, data sets, cell lines, technologies. And here's just a few that were highlighted in his paper: various antibodies, such as the MCM7, cell lines like the HeLa cell line, and also model organisms as shown here. So all these can be found along with some valuable metadata about them in our resource browser. And today's webinar, as with all our other webinars and tutorials, is being recorded, and it will pop up after we are finished with this in our website, as well with all the other YouTube videos, if you'd like to go back to it or share it with colleagues. So just a few reminders before we get started. This presentation is being recorded, and as I mentioned, it will be posted on YouTube. If you could please mute your audio and turn off your video during the presentation. And the discussion and Q&A will follow after the presentation. You can raise your hand or use the chat. Dr. Dorr is going to be discussing the cycle of cell division and death during the growth of the pancreas, and whether this is a general phenomenon in organ growth or a peculiarity of pancreas, and the significance of early postnatal beta cell death. So just to introduce today's speaker, Dr. Yuval Dorr, he's a professor of biology at Hebrew University Hadassah Medical School in Jerusalem. His work focuses on pancreatic beta cells in the context of diabetes, as well as pancreatic cancer. He has recently developed a novel blood test to study cell death in humans using methylation signatures of cell-free circulating DNA. And in terms of HIRN, he's a member of both the CBDS and HPAC consortia, collaborating closely with Drs. Kessner and Schatz as well. And so I'd like to now turn it over to Dr. Dorr. Looking forward to your talk. Thank you.
Thank you, Joyce. Let me try to share my screen. So do you see my screen, Joyce?
Yes.
You do. So thanks very much, Joyce and Laila and John, for the invitation. Happy to present in this forum, and nice to see so many familiar names and faces on the list of participants. So I'm going to talk about some insight that we got recently on how does the pancreas, and specifically the exocrine pancreas, which is most of the pancreas, grow postnatally. This is work that was almost entirely done by Dr. Miri Stolovich-Rain in my lab, which is present now, I think, in this webinar, along with other students, and Shira Anzi and Shira Azulai, and collaborators Danny Ben-Zvi and Ben Glazer from Hadassah University. And as Joyce mentioned, much of what I'm going to present was very, very recently published in this cell reports paper. And as you can see here in the simple cartoon, the question is really, how does this small pancreas at birth would grow to this large pancreas later in life? And when we talk about the pancreas, we obviously, when we talk about the size of that organ, we obviously don't talk about the islets or beta cells, which are just 1% of the story, but about the exocrine pancreas, which is the vast majority of the organ, which is composed mostly of acinar cells that, of course, are draining their content to the network of ducts. But the main component of pancreas volume and weight is the acinar cells, and these are what we're going to look at now. Right. So, what are we talking about? We're talking about this magnitude of growth. So these are simple charts that we got from either publicly available data or our own measurements. So this is how much the pancreas weight of the mouse is growing in the first month of life, from about 10 milligrams to about 200 or so. And this is a slightly different presentation. It's the increase in cell number in the mouse pancreas, but it's roughly the same as the increase in pancreas weight in the humans over the first year of life. So organ pancreas growth in the immediate postnatal time roughly follows, nothing special there, the increase in body weight. So in the first month of the mouse life, the mouse grows about 20-fold, from 1 gram to 20 gram, and the mass of the pancreas grows roughly the same. And humans, many of you may know, grow only about 3-fold in the first year of life, from about 4 kilograms to 12 or so, and so does the mass and also the cell number of the pancreas in humans. And in both species, the increase from birth to fully adult life is about 20 to 30 x. And this is what we're going to focus on now. So how does it grow? So one side issue, or not side issue, a central issue that we addressed a few years ago was the finding that when the exocrine pancreas grows, this occurs via a combination of increased cell size, increased acinar cell size, what we call hypertrophy, and increased cell number, hyperplasia. And what we found in that paper that we published several years ago was that the contribution, the relative contribution of hypertrophy and hyperplasia varies among different species. For example, in the mouse, much of the growth of the exocrine pancreas is due to a large increase in the volume of acinar cells, hypertrophy, with some increased cell number. And in humans, it's purely hyperplasia. So the size of acinar cells in the human does not change. So it's really pancreas growth is about increased cell number. And there was a very interesting correlation that we found. That was the title of that paper, that the animals with longer lifespan tend to increase their pancreas with by cell multiplication, and those with rapid growth and shorter lifespan were based on hypertrophy. But that's not the issue today. The issue today regards the involvement of cell death in that process of pancreas growth postnatally. And so to put it in some context, I think as developmental biologists, I'm typically thinking of development as composed of different stages. So there's, of course, differentiation from stem or progenitor cells to differentiate cell types, and then maturation. There is morphogenesis, right, the formation of the right structure. Then we think of post-differentiation growth, right? And this is what we expect to see postnatally. The pancreas is fully mature or is differentiated and functional. Now there's post-differentiation growth, and then there's, of course, maintenance, the wear and tear of life. And when we think about the role of programmed cell death, so I think all developmental biologists are well aware of the fact that during early stages of development, there's, of course, extensive cell death. For example, in the famous example of the elimination of the interdigit space when the hand is forming. And when you think of it, typically when we think about organ growth and maintenance, I think overall there's little evidence or little expectation that there's massive cell death. At least I thought of it as a simple process where cells either derive from stem cells or simply proliferate to generate more cells, as happens in the exocrine in the pancreas. And the question that we'll ask in the next few minutes is this the case? This is not the true story of how it all began. It began in a different way. It began with a very naive question that we asked, a very naive answer that we got. So while working on that previous dev cell paper, we were interested in seeing whether cell proliferation, because we're also studying cell growth, whether the estimations that we got for cell proliferation can fully explain the increase in organ size. So we got this large number of observations. Essentially, Miri was counting the fraction of proliferating acinar cells, Ki67 positive acinar cells, in multiple time points. In this case, is the human. So based on material that we obtained from NPOD, essentially every time point here is a section or a paraffin block from an individual that essentially has died, and the paraffin block of the pancreas is present with NPOD. And these are the fractions of acinar cells that are Ki67 positive. It's very straightforward stating. So my simple naive question was, and I won't disclose the name of the colleague that was asked to do that, can you please simulate pancreas growth based on that declining fraction of proliferating acinar cells? And the guy said, yeah, sure, here it is. It is a great mathematician. So I got a very smooth and expected result of growth of the human pancreas as a function of time in the first year. And everybody was happy initially until we noticed the y-axis that escaped our attention initially. And something here is very wrong, because the human pancreas does not grow over 1,000-fold in the first year of life. Something is wrong. And to clarify how wrong it is, this is how it's wrong, right? So based on publicly available data, the pancreas grows about 3-fold over the first year of life. And so our simulation is far, far off. And this is wrong. So to put it in numbers, the average rate of acinar cell proliferation in the human is about 2% per day in the first year of life. And this predicts this gigantic organ growth that is clearly not the case, and something is wrong with our model. And I'll be discussing in the next few minutes what's wrong in our model. But before we do that, we went back to see what this problem exists also in the mouse, and the answer is yes. So in mice, as I hinted already, the situation is slightly more complicated, because as the mouse pancreas grows, so does the size of individual acinar cells. So here are the numbers or the graphs that you need to calculate, right? So here are the fractions of proliferating Ki67 positive acinar cells in the postnatal, in the first month of mouse life, right? Going down from as high as almost 20% down to about 5% 1 month of age. As I mentioned, during that time, around weaning, the mass of individual acinar cells grows substantially, contributing to the increase in organ mass. This is the increase in pancreas weight, which is, again, in the mouse, is a function of increased cell size and increased cell number, currently due to proliferation. So when you take that into account and you simulate the expected increase in cell number in the mouse pancreas, this is what sorry, it's orange and red, but this is the expectation based on the model. While this is the calculated increase in cell number, there's actually a period in the life of the growing mouse where the number of acinar cells seems to be reduced because the size of individual cells grows so much. Anyway, there's also this discrepancy that just cannot, we need to reconcile that. So we went back to the model. How did we reach that that simulation? And there were three key assumptions. One is obviously that what we stain for, Cas67, really marks dividing cells, and not, it could be that 80 or 90% of the Cas67 positive cells in acinar cells are just resting cells that for some reason stain for Cas67. Another assumption in the model was that, taken from literature, is that cell cycle duration of acinar cells is about 1 day, 24 hours, and also the model assumed no cell death. So let's test each of these assumptions to assess the validity of Cas67 as, well, it's used by pathologists around the world, but still, does it represent only dividing cells in the growing pancreas? So we co-stain for Cas67 as well as two additional well-established cell cycle markers, CDC47, MCM2, and you can see that they correlate very well with Cas67 in multiple human pancreas samples. You can see the staining. Again, no issue with counting these cells. And the same we did with the mouse. With the mouse, we also have the advantage of being able to inject BRDU, the thymidine analog, and these are the fractions that you see. Again, a good correlation. Not surprisingly, in every sample, the fraction of Cas67 positive cells is slightly higher than the fraction of phosphohistone H3, which marks only a tiny fraction of dividing cells because it marks only mitotic cells, and also the number of it's also higher Cas67 than the fraction of cells stained with BRDU, because BRDU marks only S phase. But regardless, the correlation is such in both cases that I think it's fair to conclude that Cas67 marks only dividing cells, as every pathologist will tell you. That's how they mark, mitotic index in cancer. So assumption number one appears to be right. The second, slightly more complicated, is the assumption that, based on literature, that cell cycle duration is about 24 hours. To assess that, we injected mice with chloro-DU, followed by injection with IDU, iododeoxyuridine, at different time points. Now, if you both are incorporated into DNA, you can stain it with distinct antibodies. When you co-inject or IDU and chloro-DU, or we separate here in the top right by 2 hours, then all IDU positive cells are also chloro-DU positive cells, because almost all cells were in S phase while exposed to both reagents. However, if you separate the injections, you inject chloro-DU and then IDU, you start to see cells that are positive for one and not the other analog, allowing you to conclude that in these cells, S phase has ended by the time the second reagent was injected. Anyway, so from drawing this chart of the number of double positive cells, one can roughly estimate the duration of S phase, right? And the estimation is that the duration of S phase in acinar cells in the early postnatal mouse life is about 8 hours. So if the duration of S phase is 8 hours, now you can ask, what is the proportion of Cas67 positive cells to cells that are in S phase, which are marked by BRDU? And there are roughly about 2 to 3-fold more Cas67 positive cells than BRDU labeled cells, suggesting that the entire duration of the cell cycle is about 2 to 3 times longer than the duration of S phase. So from that, I'm sorry, I'm rushing over that, but it's pretty straightforward in that subfield. So from that, we concluded that the duration of the mouse acinar cell cycle is about 24 hours, and that's not a big surprise, because this is really what's almost all papers in literature, most normal cell types have found. So cell cycle duration is about 1 day. So this leaves us with the third assumption that there's no cell death taking place. And to address that, we took a crash course with Eli Pekarsky, the head of pathology next door, and he taught Miri and me how to identify apoptotic cells in H&E sections, and maybe more sophisticated, though less trivial, is to stain for tunnel, right? The classical marker of cell death, and which is really not trivial, is to, and Miri managed to do that, is to co-stain for tunnel and for amylase, the marker of acinar cells, so we can definitively identify acinar cells that are right now dying. There are only a few of them because of the short duration it takes. That a dying cell is exposing its, chromosome ends and is tunnel positive. Anyway, with the tunnel staining, we did manage to assess or determine the fraction of acinar cells that are tunnel positive in the growing mouse pancreas, and the number shrinks dramatically with age. But in the first month of life, there's definitely a proportion of acinar cells that are dying. Shouldn't be impressed too much by the low fraction, because cells are labeled with tunnel for a very short period. Therefore, this may mark a substantial fraction of the cells. So we conclude from that actually that assumption was wrong, and there's definitely acinar cell apoptosis during postnatal pancreas development. And when we took into account the numbers or the fraction of acinar cells that were found to be dying, with a reasonable assumption that the duration of tunnel positivity for dying cell is 37 minutes, kind of an average number from literature, and then you do again the simulation of expected increase in cell growth, you see that green curve suggesting that the discrepancy between the original calculation and real life is reduced, suggesting we are approaching to understand real life. So the conclusion from that is that there is quite massive cell death during postnatal pancreas growth, which may explain the discrepancy and may explain why there's so much proliferation and they're balancing each other. One important question that we want to ask was whether proliferation and apoptosis, since they both decline dramatically with age, are they related, the two processes, cell cycle and cell death? So to address that, we performed a pulse chase experiment with BRDU. We inject BRDU, sacrifice some of the mice shortly after, and some after 2-3 hours, and some after 1 day. And we asked two questions. One, does the number of BRDU positive acinar cells double after a chase? Which is the expectation, right? You label cells in S phase, you expect after a day to see twice the number of labeled cells, maybe with half the intensity, but twice. And the second more subtle question is whether cell death, this tunnel positivity, does this tend to occur in the BRDU positive population, which would indicate that proliferating cells are the cells that are really dying. So if you count the fraction of BRDU positive acinar cells on that, I can't recall that day, maybe day 6 postnatally, shortly after 2 hours after labeling, about 7% of the cells are BRDU positive, right? 1 day later, you would expect to see doubling of that number, and you don't see that. You see only less than 50% increase after a chase. So not all these cells gave rise to 2 healthy daughter cells. Some are missing, right? We do find them. They tend to be after the chase in doublets, as you would expect, but the total number is less than the 2-fold you would expect. We performed the experiment also in another way by fax, labeled with BRDU and count the number of BRDU positive acinar cells after a pulse and after a chase, and you see an increase, but it's not a 2-fold, and it's not a bug of the experiment, because if you do the same experiment with HeLa cells that have no issues, right? After a pulse, you get 30% BRDU positive cells. After a chase, the number increases 2-fold. So when cells divide, we know how to identify that, suggesting that indeed many of these proliferating cells or DNA synthesizing cells fail to generate 2 daughter cells. And the last another important piece of information is using that FACS sorting when you stain for BRDU in the y-axis, but also for the total amount of DNA using the HIRN marker in the x-axis, you can determine the number, the amount of DNA in the proliferating cells, right? So after 2 hours after a pulse, BRDU positive cells, which are labeled green, tend to have an SG2 amount of DNA because they're busy replicating their DNA, right? These are G1 cells, these are the G2 cells, and these are the cells that are replicating. So they are expected now to complete DNA synthesis and then either get stuck, which may explain why we don't see an increase in cell number, or divide and then go back to having normal content of a diploid cell. And this is what happened, right? So after a chase, we don't see accumulation of cells with 4N DNA. They all go back to normal, meaning that there's no cell cycle arrest at the G2M with double the amount of normal DNA, but this would suggest that those replicating cells either die off or divide to give rise to living cells with normal amount of genomic DNA. And the last, I think, piece of information regarding this experiment is that Miri was able to ask what percentage of the tunnel-positive acinar cells are actually labeled with BRDU. So in the pulse, that is animals that were sacrificed 2 hours after injection with BRDU, only 5% of dying acinar cells are labeled. So most of them do not die as they are in the S phase, but at the chase, that is cells that were in S phase 27 hours ago, right? Most tunnel positive acinar cells are BRDU positive. So the suggestion is that, right, most cell death takes place in the proliferative compartment, really supporting this co-occurrence of proliferation of apoptosis. So simple conclusion so far, right? So we do observe massive elimination of acinar cells during normal postnatal life in both or pancreas growth in both mouse and human, and this is explaining the excessive number of proliferating cells according in progress. Thank you. So we're interested in asking, how does pancreas growth affected by p53, and as geneticists, how would it be affected by the absence of p53? As I mentioned, there is actually a justification, and that's a paper, a great paper that I think often overlooked by many people. It was published 23 years ago by Steve Leach, who normally studying pancreatic cancer. In this lovely paper, he studied the model of pancreatic duct ligation. You ligate the main pancreatic duct, and as a result, there's massive spillage of acinar contents and massive acinar cell death, and the pancreas regenerates from that by massive proliferation of acinar cells. That's the normal process. And what Steve so when you do, that's the wild-type pancreas in their in one of their images. Wild-type mice after pancreatic duct ligation shows massive tunnel positivity, right, and degeneration, as you can see that. Of p53 deficiency, this does not happen, and the title is p53-dependent acinar cell apoptosis triggers acinar cell proliferation. In proliferation, also in apoptosis. But if you do the exercise in p53 null mice, you see a dramatic decrease in cell death, which is what you expect in the absence of p53, but also then a decrease in cell proliferation. So that was that paper, and essentially our question is, does p53 derive the apoptosis proliferation link also in the uninjured growing pancreas? So we essentially characterized the pancreata, the mice and the pancreata of about 1 week old, 8 days old mice. So let's go over this quickly. Body weight of these juvenile or young mice is normal. No p53, they're just normal. Pancreas weight is normal. Acinar cell size, which is a big issue in mice, is normal. Acinar cell number is normal, right? So the same number of cells, they're same size, therefore the same pancreas size. So nothing happens on first impression. But then if you look at the number of dying cells, greatly decreased, and what's most, I don't know, surprising or expected is that the fraction of dividing acinar cells in those p53 mutants is dramatically reduced, right? So from these results, we concluded that we do see p53 dependent acinar cell death. From the previous experiments, we know that this happens just prior to mitosis, and that this triggers acinar cell proliferation. So the model is that as the pancreas grows, there's a mitogen, right, that drives, to start with, acinar cells to the cell cycle, right? But many of them do not complete successfully the cell division. Many of them are sent via p53 dependent pathway. We'll talk about that in a minute, either because they suffer DNA damage or maybe because there's no trophic survival support. They are sent to die. And turns out that when they're sent to die, this cell death triggers a further mitogenic stimulus that will drive more acinar cells to enter the cell cycle. If you cut that short, there's less cell death and therefore less proliferation, but at the end you get the normal organ size, so the system is highly robust. The numbers are, I find, really striking. When we went back to the math, what turned out to be is that for 100 dividing acinar cells, the expectation would be to have the next day 200. Instead, we find only 112 live progeny. So really a much slower growth than you would expect. And so the interpretation is that the initial process there is p53 dependent, developmental, completely normal acinar cell death that triggers this compensatory proliferation. And the term compensatory proliferation is established. Stephen Leach has used that in the PDL model. It's well established in cancer, in the regeneration field, and also in early Drosophila development. As far as I know, that was not seen so far in this boring postnatal expansion by proliferation of most mammalian organs. So that's what we think is happening. Two more points that I want to make. One is when now we establish this cycle, so why does it happen? Why do acinar cells die during normal pancreas growth? They're just normal animals. Why would that happen? So we don't know. We have two models in mind. Happy to discuss and get suggestions. One is that more cells than you would think are defective, and this is sensed by someone. Maybe DNA damage makes sense in light of p53 being involved there. And if many of the cells are eliminated, this would cause that phenomenon of cell death responded for by compensatory proliferation to achieve the right total cell number eventually. And you may think of that as a quality control mechanism to ensure healthy genomes that may help that individual live long and prosper. A second different model is that this may relate to morphogenesis. So what we really speculate is that when the pancreas grows, the number of acini is increasing. And the process of new acinus formation is not fully understood. It is not trivial. It's not just expansion. If that acinus wants to give rise now to a new acinus, acinar cells here have to divide, actually move away and find another end of a ductule and rearrange around that. And so we speculate that perhaps there's some trophic interaction or trophic support of acinar cells by the terminal cells of the duct, so-called central acinar cells, that ensures the proper functional morphology. You don't want an acinar cells be stuck in the wilderness and spritzing the contents not to the ducts. In a shorter version, perhaps we should think of postnatal pancreas growth as definitely a morphogenic process that does involve, therefore, the classical cell death during organization of a new organ. We don't know what's the reason for cell death. Maybe there's another model. And finally, is the question that is still remains open, whether this is a general phenomenon. I'll just mention that in our favorite other cell type, the beta cells, there is actually a known documentation of a wave of beta cell apoptosis during early postnatal life. This was shown by both initially by Susan Bonner-Weir, apoptosis during remodeling of the endocrine pancreas in the neonatal rat, and then Ben Glaser showed that this happens also in the, I think it was the first year of human life. So this does happen, and I think the interpretation of that is not fully understood. Perhaps this has to do with that process that we've seen in the exocrine pancreas. And beyond that, we're not really sure. We think we have evidence that this happens also in the context of liver postnatal development, but we're not sure yet. I personally find it hard to believe that this is a unique phenomenon in the exocrine pancreas. And I'm sorry, when we simulate from the little bit of data that we have, we simulate the expected increase in mouse beta cell number. This is what we get. So, and again, a discrepancy between simulation and actual growth, suggesting that there is really much more than you would need beta cell proliferation balanced by, or maybe triggered by, massive beta cell death. Last, a few thoughts on what is the significance or the implications of that extensive apoptosis and coupled proliferation that we see during pancreas growth. So one concern that always comes up is, why would nature act with such a waste? So I want to preempt that question and say that I don't think there's a waste, because we think just wrongly about energy metabolism in the growing animal. And I think actually the pancreas doesn't care if you eliminate a cell, because the contents of that cell would be recycled. So I think when we do the economy of cell death, birth and death, I don't think that waste is a consideration. A second potential implication is that if you want to move from X cells to Y cells in a given organ, pancreas or maybe otherwise, there's this concept of mitotic clocks, right? How many cell divisions take place, and we roughly know how many mutations occur in every cell cycle. So there are these mitotic clocks. But if the actual number of cell divisions with cell death is way higher than you would think, then the expected number of somatic mutations should be recalibrated. And I think that's an important consideration. People think about somatic mutations. A specific prediction for the p53 mutants, which, mind you, Li-Fraumeni syndrome, there are people that are born with p53 heterozygosity, so that's not far from reality. So in the p53 mutants, it seems that those damaged astronal cells that were destined to die actually survived. So perhaps if these are cells that suffer DNA damage, then the expectation is that in those p53 mutants, we'd expect accumulation of somatic mutations in the adult, in their adult pancreas or maybe other organs. And surprisingly, as far as I can tell, this was not tested prior to the onset of cancer. Two more points about cancer is that actually it's known that in the context of cancer, much or if not most of cell proliferation is actually balanced by cell death, which explains slower growth, right? When someone is diagnosed with cancer, even if the pathologist in the biopsy is in the biopsy sees an invasive cancer and finds a dramatic or not dramatic proliferation rate of, I don't know, 10%, right? You would expect that daily 10% proliferation to lead to doubling of tumor mass in 1 week. So you should rush to the surgery room, but that's not the case, right? We know that that tumor will grow very slowly. So this is a known phenomenon, but I think most people think of that as a cancer-related phenomenon, but perhaps this is actually normal physiology and not a cancer-specific trait. Another point that has to do with another thing that we're doing in the lab, and that is liquid biopsies that actually are looking for cell-free DNA coming from dying cells, and nobody knows who are these dying cells, and it's possible that cell-free DNA from cancer cells and maybe in other pathologies is actually not a general reflection of the randomly selected cells that died off and released their cell-free DNA, but specifically the proliferative compartment of, let's say, tumors that are also dying. And this means, I think, important things in the liquid biopsy space, because it would mean that what you learn about cancer from liquid biopsy is only about the proliferative compartment. And then there are also thoughts about the physiological significance of that process in general, which I think have to do with robustness. If you think about that, when if there's a mechanism for both regulated cell birth and cell death, you become much more robust for perturbations. And here's one big perturbation: you lost p53, cell death rates declined dramatically. You would expect to get a giant pancreas, but you don't, because of this link. So you perform excessive rounds of cell cycle and cell death, but this gives the organism flexibility to resist perturbations. So I think I'll stop here, and happy to take questions and discuss.
Thank you very much. That was a very interesting speech. Really appreciate it. Looks like Olivier has a question.
Olivier.
Olivier?
Joyce, I think we might, let me unmute him. Hold on.
Yeah, Leila, can you unmute everybody now?
Speakers should be able to unmute themselves. Olivier, can you unmute yourself?
Apologize, we lost internet a couple times there. That's why the recording
Oh,
stopped.
sorry. Yeah, thank you, Yuval. That was beautiful. I have a liquid biopsy kind of question.
Yes.
So a few years ago, you tried, right, to use circulated methylated DNA to assess beta cell loss, and it works to some degree, but the loss is so small that you reach an issue with sensitivity. So my first question is that also the case if you try to measure acinar loss in the neonatal space? And a related question would be, is that something that we should try in young children who are at least developing type 1 relation between acinar death and risk of type 1D?
Yeah, thank you. Very good point. So you're right that we have very powerful tool for identifying beta cell DNA in the plasma, and when it's there, we see that. We see that mostly in the context of islet transplantation or pancreas transplantation, but in normal life, as well as in children with type 1 diabetes, developing type 1 diabetes, we don't see that because there's nearly zero. And the reason is that the assay is not sensitive enough, but there's also an issue, and that is that the fact that when cells die, most material from those dying cells is actually consumed locally, and only a small fraction reaches circulation. In the context of beta cells and type 1 diabetes, that fraction that reaches circulation is not sufficient to be detected. In the exocrine pancreas, which is, of course, 100 times bigger, it's easier to see cell-free DNA. So I don't know about early postnatal life. It's a great suggestion. Hard to get plasma samples from young children, but it's a good suggestion. We did look a bit on plasma samples from children that were developing type 1 diabetes and looked for acinar cell markers, and we didn't see much, but we can do much better there. What I can say is that the detection of acinar cell cell-free DNA is a big thing in diagnosis of pancreatic cancer. And this is how people think the best way is to identify pancreatic cancer early. Multiple applications on that, even from us. In early stages of pancreatic cancer, you see elevated levels of acinar cell-free DNA, also in pancreatitis. Whether this can be identified in the early stages of T1D, good experiment.
Thank you. Ernesto, did you have a question? You want to unmute yourself?
Hi. Hi, Yuval. It's quite a nice talk. And, I have a more fundamental question, about the cell biology, because from what I understand, the mammalian cells, they are symmetrical when they divide, but there are a lot of microorganisms, for example, that when they divide, you have a parental cell and a daughter cell. They're not symmetrical. Does mammalian cell have any kind of mechanism for checking the symmetry, and they say that some organelle is not well divided in the two daughter cell? Would that trigger some kind of cell death too?
It's truly a basic question. So first of all, there is, of course, such a thing as asymmetric cell divisions in mammals, and this is typically when you talk about stem cells or progenital cells, when they divide and one daughter remains in the niche and remains a stem cell, and the other daughter becomes differentiated. As far as we know, in the growing exocrine pancreas, organ growth occurs via division of fully differentiated cells, postnatally, no involvement of stem cells or progenitors. And as far as we know, when an acinar cell divides, if everything goes fine and there's no cell death, you get two healthy, identical daughter cells. There are, of course, checkpoints that are meant to check just prior to division that everything is fine. I'm not aware of mechanisms that check whether the two daughter cells are also, containing everything they need, but maybe this is why some of them die. So we mostly think of differentiated cell divisions as symmetrical, and the checkpoint is typically just prior to mitosis, G2M checkpoint.
Right. Thank you.
Great. John, did you have a question?
Yeah. First, thank you, Yuval. When you were your preemptive strike on the waste question, I was laughing because that's exactly what I was thinking. So thank you for answering that. Yuval, my question is, so you presented two potential models, which are interesting to talk about why this is happening with acinar cells. My question is, are all acinar cells the same?
Well, big question. I think there's mounting evidence that there is heterogeneity among acinar cells. I think it's a super interesting topic for further study. I mean, very few serious studies have addressed that. I mean, on first approximation, they're all identical. They're all sacs sitting around a central acinar space, spilling digestive enzymes, and that's it. Nobody so far was able to identify a proliferative compartment among acinar cells or differential functions of different acinar cells. So there are hints coming typically from recent single-cell RNA-seq data, but we don't really know of functional heterogeneity, and we don't know about replicative heterogeneity. So I guess null hypothesis is that they're all the same, and it remains to prove otherwise.
It's interesting what Olivier was asking, and you sort of suggested it, because with the exocrine pancreas, you might be able to see something in circulation. And I know that you have looked at EVs in the past, and what could a whole bunch of apoptotic EVs look like from acinars, and could they be different? I don't know. I'm not even sure if those are legitimate questions, but it seems interesting. I
agree. It's interesting. I think the field is moving towards more and more sophisticated, non-invasive technologies to assess cell death using material in plasma, and there are ways to, from my other life in liquid biopsy space. The simple thing is to say, well, here's DNA coming based on DNA methylation patterns coming from that cell type, suggesting that this cell type has died, and I can quantify that. But there are now technologies that actually allow you, from looking at the DNA fragmentation pattern, etc., to conclude not only which cell this came from, but also what was the gene expression pattern in the cell before it died and released that cell-free DNA. So big things are expected in the future.
Very tricky. Yeah, thank you, Yuval. I appreciate it. Nice presentation.
Thank you. Ken, did you have a question? Ken, did you want to unmute yourself? Maybe he doesn't know he has his.
Okay, yeah, no, it finally let me unmute. Thank you.
Okay, good.
Thanks, Yuval, for a nice talk. So many years ago, in 2006, we published a paper looking at the effect of FLIC1 mutation on pancreatic induction, and looked really hard and found, I rarely say zero, but really zero apoptosis in the endoderm during pancreatic induction. But your data is very compelling. And what I was wondering is, getting to your point about how it's a mature acinar cell that's dividing, maybe because of all of the nasty enzymes in those cells, they're particularly sensitive to cellular damage, not just DNA damage detected by P53, but all kinds of other stuff. And as you go through mitosis and there's nuclear envelope breakdown and et cetera, just the cells are more inherently sensitive to all kinds of damage. And so I like the model that you're studying, but I just want to put out there that perhaps the special metabolic capacities of the acinar cells are part of what make them so fragile in your context.
Thank you. I think it's a very good thought. They are packed with highly dangerous material, and we do know that they don't take precaution measures. They don't de-differentiate before dividing. They actually divide with all these sacks of dangerous enzymes. So it's an interesting idea that may give some rationale in the case that this phenomenon remains limited to the exocrine pancreas and does not expand to other growing tissues, which will be tested empirically. I mean, maybe I should have stressed that we know of cell death events taking place during development and morphogenesis, but there are definitely contexts in which a cell type or an organ or a tissue is dividing rapidly and increasing at a rate that is expected based on the number of dividing cells. Right, in embryonic stem cells in vitro, you see multiplication, also tissue culture cells, and also in other contexts. For example, in our favorite is liver regeneration. Right, if you perform partial hepatectomy, one can calculate the time to regeneration to normal liver mass based on the number of dividing hepatocytes. There does not seem to be much of cell death during that rapid physiological response, but when the pancreas grows, something goes wrong, and your suggestion is very interesting.
Thank you. There are a couple questions in the chat as well. Let's see if we can look at them. What do you think about Birdute positive cells labeled in very young pancreas that could also be LRC label remaining stem cells, which divide rarely and will not provide progeny?
Yes. So I failed to mention in the introduction, and that was also a question of one of the reviewers. As far as we know from lineage tracing studies, and we actually reproduced that again for the purpose of that experiment, postnatally, there are no stem cells in the pancreas. There are no label retaining cells. All you've got is fully differentiated endocrine cells and fully differentiated duct cells and fully differentiated acinar cells, and each compartment is on its own. And as far as we know, growth is via division, cell cycle, division, and proliferation of differentiated cells. So lineage tracing does not suggest that such label retaining stem cells do not exist. There's Shall I read that,
Yeah,
Joyce?
there's another question. Yes.
Yeah, Daniel Dean, could you comment on the turnover in respect to the cycling between normal physiological state, for example, fasting versus feeding, for example, availability of nutrient signals by mitogen, biomass, CCK? Fantastic question. The pancreas responds dramatically to fasting. It shrinks. In the mouse, it shrinks twofold. I think in humans as well, and this goes along with the atrophy of the intestine. You don't eat, you you lose about half of your GI tract, including the pancreas, which has nothing to do when you don't eat. And this regrows again after feeding via a combination of cell growth, hypertrophy, and cell proliferation. Much is known about that, much remains unknown about that, and what is mentioned here, mTOR, CCK, is exactly the things we're interested in. There are specific signaling pathways that are responsible for the feeding-related regrowth of the pancreas. But our mice were suckling continuously, so this is not probably the case, but it's an interesting phenomenon.
Thank you. Maybe we have time for one more question from Janelle.
Oh, hello, Jeanette.
Can you unmute?
Okay. I had two questions, Yuval. That was a really great talk. It's really interesting stuff. So you mentioned this model where you have proliferation triggering or cell death triggering proliferation. So is there a spatial relationship between the tunnel positive cells and the BRD positive cells or the proliferating cells?
Yes, they're the same as far as we can tell. So the dying cell, that's a good question. And one possibility was that when cells divide here, they die there or in adjacent, but it seems to be a cell autonomous phenomenon. So I showed a slide before showing that most tunnel positive cells are actually BRDU positive in this pulse chase experiment, suggesting that most cell death takes place in the compartment of dividing cells, and we think that this happens right before mitosis. Actually, when p53 is known to kill cells.
But I guess I thought about that model where you see that the BRDU cells tend to be more tunnel positive, but that would suggest that the proliferation comes before the cell death, instead of the cell death triggering the proliferation.
It's it's definitely a cycle, right? It's not a linear process. There's certainly a mitogenic stimulus that starts all growth. There's certainly cell death that is primarily affected by p53 because that's what p53 does. And then this refeeds to the mitosis machinery. So what comes first? I think that when the pancreas grows, my sense is that what comes first is a mitogenic stimulus. By the way, in adult life, when you're in a homeostatic situation, right, my pancreas does not have to grow now. So I would think that the typical trigger would be cell death, which is then compensated for by proliferation. But when you grow, there's net expansion.
Yeah, that's why I thought there might be a spatial relationship where a cell is dying and then like a nearby cell is more likely to be proliferating.
So we don't see that. Now I see what you mean. So no, what you're saying is that even if the dying cell was in the business of cell cycle, the compensating cell that would proliferate perhaps resides next door and not far away. Excellent question. We haven't looked at that. Should have. Good point. Thank you.
That's a great point. Okay, thank you very much. I think that's all the time we have. Thanks again for a terrific talk, Dr. Dorr. We really appreciate it. And remember, this will be posted on our YouTube channel on our website very soon. Thank you all for attending and hope to see you again at a future webinar. Thank you.
Thank you. All right.